Hello, my dear students. I'm Dr. Vaishaly Bharambe. I've been teaching anatomy for more than 25 years and I love it. I know many of you have just taken admission in your MBBS and you've been just taught cell. There's a lot of information they're giving you, and sometimes when you open the book, you find that much information is given in the book and much more as well. How to study, how much to study becomes very difficult to understand. So today what I'm going to do is give you a quick brief on cell in the form of cell part 1, and there's one more lecture next, cell part 2. In these two short videos, I'm going to try and cover important aspects of the cell that you should know while moving forward in the subject of histology. So let's start by defining the cell. Cell can be defined as the structural and functional unit of the body. You were taught this in your school, you know it. Now you know that your body goes through a lot of functions daily. Ingestion, digestion, absorption, reproduction, and so on, excretion. A cell goes through all of these too. So you can imagine that your cell is like your body. You will feel yourself familiar with it. Okay? Cell, like we know, is 70% water. The rest of it is organic and a lot of inorganic material. It has variable shapes to it. It has variable sizes to it. I'm not discussing this because I know you know it. It was taught in your school. A few questions to start you off. Which is the largest cell of the human body? Which is the longest cell of the human body? Think and get back to me. Please write the answers in the comment section. I would love to know. Right. Remember, if I've asked you a question, my video will always give you an answer as well. So the answer will be found in the answer section or in the notes on my website. Okay. So feel free to look for the answer there, or of course feel free to message to me. Now, I want you to draw this cell, if possible. I have drawn it. I want you to draw this cell. What is it that I want you to draw? I want you to draw first the nucleus as seen here. Remember, a cell is made up of cytoplasm and nucleus. This here is the nucleus, which consists of a fluid content called nucleoplasm, some thread-like material called chromatin, and a dense body in the center called nucleolus. It is surrounded by a cover or nuclear envelope which has got holes in it which are called nuclear pores. Through these holes, the nucleus communicates with the surrounding cell. Now this is the cell covering or the cell membrane, and this is the cytosol, the content of the cell beyond the nucleus. Okay. Now, you got this much? Let's move on now to cytosol. What does the cytosol contain? Cytosol is basically a lot of fluid and has got a lot of other functional organelles in it. Let's take a look. You have organelles such as rough endoplasmic reticulum, mitochondria, which was taught you in your school, centrioles, the body that helps the cell multiply, a peroxisome. Also contains smooth endoplasmic reticulum, lysosome, secretory vesicles, Golgi apparatus, free ribosomes. Now this is your cell membrane. You can see how the cell membrane has folded to form microvilli and cilia. All this is the cell, and that is the reason why you find it so challenging, isn't it? Now further, on inside of that cell you find the cell skeleton called as cytoskeleton. So you as a student are expected to know this all. And as a new student in medicine, you often find it too challenging. So I'm going to cut it down to small bits by starting our study by discussing the nucleus. Okay, so that's the core, the center of the cell, the nucleus. So that's why you're going to study nucleoplasm, nucleolus, chromatin, and so on. So nucleus, an essential component of any cell. Its appearance in the cell is usually basophilic. It often looks dense and basophilic, bluish in color. It can be round, oval, flat, rod-like. It can be central, it can be eccentric, it can be placed a little basally in the cell. Please observe all this. When you go and start observing under the microscope, you will see all this. Okay? And the size of the nucleus is also variable. So, shape is variable, size is variable, position is variable. Okay, right. Now, number, is it variable? Can a cell have more than one nucleus? Yes, sometimes it can. For example, here you are seeing a section through the urinary bladder, transitional epithelium. You are seeing there are two nuclei here. So, some few cells in the body sometimes have two nuclei. Liver cells also sometimes have two nuclei. Sometimes, for example, your skeletal muscle, multinucleated, has so many nuclei. So this happens rarely. Normally, there is one nucleus. Now, question again. Can you name a cell that does not show a nucleus? I'm sure you can. Anyway, write the answer in the comment section, or you will find the answer in the notes section on my website. Yes, you know this, red blood cells. So what is an interphase nucleus? Till now, we got some basics of nucleus now. I have a question for you. What is an interphase nucleus? Interphase nucleus is a nucleus that is functional, but the cell is not in division phase. Nucleus is functioning, okay, but cell is not actively dividing. So here the nuclear membrane is well defined. The chromatin material is spread out. What is the other phase then? The other phase is a mitotic phase when the cell is actually in division. In this, you will not find a nuclear membrane and you will find chromatin material in the form of chromosomes. That is the difference between an interphase nucleus and a mitotic nucleus. Right. So what are the parts of nucleus? So much, so many things. Why bother to read when we can see them? Always remember, you should have visual representation of anatomy, makes learning much easier. So here you are seeing a nucleus surrounded by a nuclear envelope. So I want you to focus on this, okay? So here is your nuclear membrane or nuclear envelope. You can see that it's got holes in it. Here, here, here. It is through these holes that the nucleus communicates and controls activities within the cell cytoplasm. Here is the nucleoplasm, which is a colloid fluid material in which the chromatin threads are spread out, in which the nucleolus can be found. Now, terms to remember: outer nuclear membrane, inner nuclear membrane. Nuclear envelope is having two layers, outer and inner. And in between them, it has got perinuclear space, space between outer and inner nuclear membrane. And these are the nuclear pores, like we just discussed. If you were to go further inside, you find that the nuclear pores are guarded by a nuclear pore complex. So, it's not that we need to know this, but we should know that the passage into the nucleus and out of it is also controlled by something, nuclear pore complex. The nucleus just below the membrane has got something like a cytoskeleton, which is called nuclear lamina, which gives structure to the nucleus too. Okay, so that's some basic terms that you should know. Now in the center or on the edge of the nucleus is a dense body called nucleolus. Okay, this is the site of ribosomal RNA synthesis. The RNA will leave from here through the nuclear pore and bring about protein synthesis in the cell. So the nucleolus is a metabolically active site of the nucleus. You can have one, you can have many nucleoli. Maximum observed have been 5. Okay, so that's the nucleolus. Now let's come to something again taught you in school, but I'm going to take you through it once. Here is a chromosome. Okay, I want you to just pause. Suppose this simple ball of wool is chromosome. As you begin to unravel it, okay, what happens is you start to see chromatin threads. If these threads are spread out, you call it euchromatin. But if they are clumped together like this, you call it heterochromatin. Euchromatin, heterochromatin. Heterochromatin will be clumped up chromatin. Okay. Suppose you have euchromatin and you further expand it, you begin to see chromatin fiber. Further expanded, you finally come to chromatin string. And you further expand it, you come to see a DNA double helix of DNA molecule. Got it? So this is very simply the concept of DNA to chromosome or chromosome to DNA. It's just a matter of coiling and uncoiling. Okay? But we learned some terms here. We learned that when the chromatin is very uncoiled, you call it euchromatin. And such a nucleus looks very light stained and is called as euchromatic nucleus. Okay? It indicates RNA synthesis. It's a feature of a very active cell. When the cell is very active, the nucleus tends to stain very light. Okay, it will have a prominent nucleolus and is said to be open-faced nucleus. Open-faced, it's not closed, it's open-faced. Whereas, think of the clumped wool, heterochromatic nucleus. The chromatin material is very clumped. It stains dark, may not show the nucleolus very well. It is indicative of an inactive nucleus. It's called as a close-faced nucleus. An additional term you may come across is called vesicular nucleus. Vesicular is even more active. It's much lighter. Nucleolus is very prominent. Okay, it's indicative of a high metabolic activity of a cell. Sometimes you find the nucleus is pycnotic. Nucleus is shrunk, misshapen, dark, basophilic, looks degenerating, is degenerating. Nucleus of a dying cell is called as a pycnotic nucleus. You will come across this term later when you begin to study pathology. So 4 types of nuclei you saw: euchromatic, heterochromatic, vesicular, pycnotic. Try and understand the concept. Don't go too much into details. Right. So now we've got a brief introduction to the nucleus. Let's go out and see the rest of the world. Okay. So nucleus, and then there is the cytoplasm surrounding it. Cytoplasm contains cell organelles, cell inclusions, cytoskeleton to give it shape, and finally an aqueous gel in which all this happens. Okay. So what is protoplasm? I keep asking you questions, hoping that you will tell me the answers or write them in the comment section. Remember, all answers are always there on my website. You only have to go and access them there. I don't ask you a question to which I won't give you an answer. Right. So cytoplasm. Cytoplasm, like I told you, contains cell organelles. What is a cell organelle? Cell organelle is a little organ. A little organ in a cell is called as a cell organelle, which performs specific active function in the cell. Then what is cell inclusion? Somebody who is there in the cell but is performing a passive storage job. It's just storage, passive job. It's not actively performing any action. So that is called as a cell inclusion. Let's talk about cytoskeleton. Common sense, something that gives shape to the cell is called as a cytoskeleton. And cytoplasmic matrix is the aqueous medium in which all this is happening. It can contain proteins, enzymes, small molecules, etc. We will discuss it at some point in our discussion. So, take a look at this image now. This is the cytosol or the cytoplasmic matrix. These are all the little organelles that I talked about. Some more, some more. This is the cell membrane, microvilli folding into microvilli and cilia, and this is the cytoskeleton. Now we are going to talk about all of these in brief. So let's understand that these organelles that I've been talking about are divided into membranous and non-membranous. What is this? So see, what happens is these organs often have a membrane around them so that they have an inner environment which is different from the environment within the cell. They are helping to create microcompartments in the cell. I think this is best understood with the help of an image. You are seeing a cell here. You are seeing this smooth endoplasmic reticulum surrounded by a membrane. What is happening in this smooth endoplasmic reticulum is different from what is happening in the rest of cytosol. Same thing for the rough endoplasmic reticulum. Same thing for the Golgi complex. Same thing for the transport vesicle. So inside the organelle, the environment is different from the environment in the rest of the cytoplasmic matrix. Thus, microcompartments are created by these membranes in a cell. Such organelles are called as membranous organelles. Question: what is vacuoloplasm? What is hyaloplasm? Find out and let me know. Again, you will find the answers on my website or in the notes. So now let's classify our cell organelles into membranous and non-membranous. Okay, this is there for you to peruse. I won't be reading it. Let's go on to the first structure we want to talk about, which is the plasma membrane. There is a note here. Please pause the video and read it. I would like to move on. Cell membrane or plasma membrane, the covering of the cell. So, here is the cell membrane. This is what I want you to ponder upon. The cell membrane is made up of lipids, carbohydrates, and proteins. Lipids are there in the form of such lipid molecules called phospholipid molecules. This one with the two tails, okay, is a phospholipid molecule. So, there are 1, 2, 3, 4 phospholipid molecules in this image. Okay. This is one phospholipid molecule. Now, this is the plasma membrane or cell membrane. Here you are seeing the phospholipid molecules, but here you are seeing something else, the protein molecules. As you can see, the protein molecules can be present throughout cell membrane, can be present only on the outside, can be present only on the inside. Okay, so protein molecules are present, and then on the top, on the outer surface, you have attachment of carbohydrate molecules. They can be attached to protein, they can be attached to the lipid. Okay, so lipids, carbohydrates, proteins. So what type of proteins? Like I said, there are proteins forming the structure of the plasma membrane, proteins performing certain functions. Okay, so what functions? They can perform they can form the structure of the plasma membrane. They can act as receptors. Some hormone is secreted. It can come and get attached to a protein to act on the cell, or act as enzymes, or act as carrier protein. You can have the protein binding with something, here as you can see, carrying it across and releasing it in the cell. Or the protein is present throughout the cell membrane and creates a tubular channel through which material is entering into the cell. This is called as a passive channel. So all these are functions of your proteins. Similarly, carbohydrates can bind proteins to form glycoproteins, can bind lipids to form glycolipids. Okay. They can coat the cell to form what is called glycocalyx. The glycocalyx helps to hold the two cells together, helps to create a negative charge around the cell, repelling other structures, gives an antigenic property to the cell. All these are functions of carbohydrates. So, what functions does a cell membrane do? We saw that cell membrane is made up of lipids, proteins, and carbohydrates. What functions does it perform? It performs the function of giving the cell a shape. Yes. It has underlying cytoskeleton attached to it, further helping to maintain shape. Okay. It is responsible for control of passage of substances in and out of the cell. How? Let's try and understand. It also has a receptor function. It also has a sensory function. Now I want you to focus on passage of substances into and out of the cell. This is easy and this is fun, believe me. So we are talking about the process of endocytosis and the process of exocytosis. Ready? All right, take a deep breath, drink some water, relax. Let's get into this very interesting part of the cell. So transport of material across the cell membrane is called as endocytosis. Remember, I told you everything that you do, a cell does. You drank water, so does the cell. You ate something, so does the cell. Everything the cell does, everything you do, the cell does. Right? So you can drink fluid. Okay? That drinking fluid or cell drinking is called pinocytosis. And what is formed is called pinocytic vesicle. Now let's go for a meal. The cell wants to eat. When the cell eats, it's called phagocytosis. The vesicle membrane-bound structure that is formed is called phagosome. Right? Let's take a look at how this happens. Here are some molecules who have approached the cell. They want to enter into it. Here is the cell membrane. The cell membrane invaginates, just dips down, allowing that material to enter inside. As the material enters inside, the cell membrane fuses off again, and the material is in the cell, forming an endocytic vesicle. Now it's floating in the cytosol. That may contain fluid, in which case you will call it a pinocytic vesicle. That may contain some solid matter, in which case you will call it a phagosome. All are endosomes or endocytic vesicles. If you have eaten, you also need to go to the loo, so does the cell. Okay? So the cell also wants to excrete, get rid of some material. Cell also wants to secrete. Sometimes you produce something that you want to give to somebody. So does the cell. So now here, this process is called exocytosis. Removing something from the cell, either for process of secretion or for process of excretion. So here is a vesicle which has approached the cell membrane. I want to release something outside, right? Secretory vesicle. It approaches cell membrane. The vesicle fuses with the cell membrane. See how they're just fusing with each other. The cell membrane now opens out, releasing the content. Process is called as exocytosis. Okay, so cell eating, cell drinking, cell are called endocytosis. Cell releasing material is called exocytosis. Okay, in either case, there are membrane bound vesicles involved. For exocytosis, there was a secretory vesicle. For endocytosis, there was formation of pinocytic vesicle and phagosome. Right. So we got quite a bit of information about cell membrane. Here is the cell membrane which is involved in all this. This is how a secretory vesicle has started to approach the cell membrane. You can see them fusing, releasing the contents outside by process of exocytosis. This is a broader picture of what we just discussed. Now understand that not only cell membrane is involved in endocytosis and exocytosis, it is also involved in holding different cells together. Every cell cannot go its own way. They have to function as a unit, isn't it? So you have to hold cells together. These methods by which you hold cells together is called as cell adhesion. Today is not the time for discussing cell adhesions, but I am giving you a broad classification along which you can explore. Soon I'll create a video and release a video about cell adhesions. That's when I will be exploring all this. But do not think that you can get away by not knowing the basics about it. There are unspecialized cell adhesions, there are specialized cell adhesions. Specialized cell adhesions can be divided into anchoring junctions, occluding junctions, and gap junctions. What are these? Think about it. And finally, there are junctional complexes. A mix of all of these are called junctional complexes. Leaving you with a question of cell adhesions, I will complete this lecture, Cell Part 1, in which we learned quite a bit. But we have a long journey ahead. Cell Part 2, we will be discussing all the other organelles in brief. Okay, in brief. I cannot go into details because then what's the difference? The books are giving you details. But to understand the concept is important. Then you can individually explore details from books. So this was Cell Part 1. We've finished our discussion on nucleus and cell membrane. We will now be diving into the cytosol. Alright, let's see you in the next video, Cell Part 2. Meanwhile, if you like the lecture, do like and subscribe to my channel. Students, do pass comments in the comments section. I need you to do that to make me understand whether my videos are really helping you or not. Do you need something more from me? Do you need me to shorten them, give you more details? Whatever is the requirement of a student, my VB Anatomy is ready to meet it. But I need to know what your requirements are. So I hope you enjoyed this lecture. I'll see you across the screen in my next video, Cell Part 2. Bye.